First, it's my pleasure to introduce Dr. Tilman Buerckstuemmer. He's the head of research for Haplogen. He'll be spending most of the presentation talking about the technology and background of the organization and the value that their products can provide to you. I'll be closing out the presentation at the back end, talking just a little bit about what the commercial offer is and how to access their cell lines and clones through us. And with that, I will pass the presentation over to Dr. Buerckstuemmer.
Then welcome everybody. It's a great pleasure to be here this afternoon with you guys. So I thought that what I was going to start with is by reminding us that mutations have yielded very important insight in various areas of biology. For instance, the entire cell cycle was revealed using yeast genetics. In flies, a lot of the knowledge that we have today about developmental biology has been found in flies, and the entire apoptosis pathway has been worked out in yeast. And really, the reason why we cherish those model organisms is because we have genetic tools at hand that allow us to study those pathways. Yet when it comes to mammals in particular, we're either studying mice, where we're left with homologous recombination, which, as you know, is a tedious and costly process, or until recently, when we were studying genes in human cells, we had to use RNA interference. And the problem with RNA interference is that in many instances, we only get incomplete gene inactivation. On top of that, and that is especially a problem if we're looking at larger scale screens, RNAi is poised with off-target effects, and as a result, the results that were obtained with RNAi are considered not entirely reliable, and that is just illustrated in that screen where we're looking at 3 RNAi screens published in very renowned scientific journals, and they screen for host factors of HIV. They each find round about 200 to 300 human genes, yet the overlap between the 3 genes, between the 3 screens, is really 3 genes, which is almost random. And so what we would really like to have to be able to do genetics in human cells is a cell line in which we could make genetic alterations. And the problem that we and others have run into doing so is really a consequence of the fact that humans are diploid, so they have 2 copies of every gene. And the consequence of this is that if we inactivate one gene copy, the second gene copy usually stays intact, and that is sufficient not to elicit a phenotype, so the mutation is usually masked by that second intact copy. And so then very recently, Tijn Brommelkamp, at the time at the Whitehead Institute and now at NKI in the Netherlands, came across this human cell line which is called KBM7, and that is a cell line that is haploid for all chromosomes with the exception of 8, and we see here a karyotypic or a karyotyping that essentially stains every single chromosome. We see 8 is diploid. We see that cell line has the Philadelphia chromosome. This is a translocation between 9 and 22. But apart from that, it's actually a fairly normal and well-defined and karyotypically stable cell line. And he used that cell line for a seminal paper published in 2009 in Science, where he actually started doing genetics in that particular cell line, and that is the cell line that we're going to use for the experiments and the tools that I'm going to describe to you for the rest of this presentation. So let's have a look at, what is that cell line? So first of all, we think the exciting prospect is that this is a human cell line. It was originally derived from the chronic myeloid leukemia patient, so it's a blood cell, it's a suspension cell, as we see in this image. As mentioned before, it contains the BCR-ABL translocation, yet we've done extensive characterization of that cell line at the genome, the transcriptome and proteome level, and we find very few additional mutations. There is a point mutation in P53 and one in Notch, but apart from that, this is a fairly normal cell line. They grow easily and rapidly in cell culture. As mentioned before, they're quite well characterized, and all of this information is available through a tool, and I can show you, And I can show you the website again at the end of the talk. This is a tool where all these genomic and proteomic information can actually be publicly accessed. Okay, so then in those cells, we no longer have that problem, right? Because we have, this is the problem we had in diploid cells. Genetics doesn't really work. If we have a haploid cell, the mutation immediately translates into a phenotype. So then you may wonder, how do we introduce those mutations? For that, we actually use a principle that is well established in obtaining heterozygous mice, so-called gene-trap mice. This is a technology that's probably 15 years old by now, so it's well established. And the way this works is you actually use a retrovirus to insert this element into the genome, and if this element lands, let's say, between exon 1 and 2 of a gene, it terminates the transcription of that gene at that particular locus. How does this work? We have, in this cassette, we actually have a splice acceptor GFP element here that will trap the splice donor from exon 1. I'm sorry, there is something wrong here. The splice donor from exon 1, so that we get a marker gene GFP expression. And then, because we have a polyadenylation signal present in this cassette, we will terminate transcription right after where the gene trap inserts itself. And so this is a tool that we can use at quite high throughput to actually knock out individual genes. The downside is, and I'm pointing this out here already, is that we cannot choose where that gene trap cassette inserts. So this will essentially insert where the retrovirus that we use as a vehicle decides to land. And so now, because we can produce very high titer retroviruses, we can actually, for the first time, do unbiased genetic screens in human cells, and this is how this would work in principle. So we start off with a large pool of cells. This is a pool that may contain as many as 10 million or 100 million cells in the first place. I'm sorry, there's something wrong with that. And we can mutagenize them using the gene trap at very high coverage so that every gene is affected multiple times in that pool of cells. We can then subject them to a selection, and that is something that I can describe to you in a moment, what we can actually use here. And then we can investigate by deep sequencing where did the gene trap land. So what are the gene trap mutations that give an advantage in that selection procedure, and we can map them back through the human genome. And so the easiest selection scheme that we can imagine is a positive selection where we use either bacteria or bacterial toxins, viruses, or small molecules, and all of those would have in common that they kill the majority of the cells, and they really leave us with the few resistant mutants that no longer respond to the selecting agent. And so I would like to show you one example of how this works. Sorry, this is really disturbing. Okay. Let me show you one example of how this works. This is a published example in which people looked for a receptor for the Clostridium difficile transferase. This is a toxin of a bacterium that is really causing some nasty disease, particularly in hospitals. And the question was, what's the cellular receptor for that toxin? So the way we, and especially Time Brummelkamp, who did these experiments in his lab, addressed this question is we start off with a bottle of mutagenized cells. Again, these contain many, many independent mutations, and they cover every gene multiple times. We then expose them to Clostridium difficile transferase. So most of the cells will die, but a few cells, and let's hope so, also the cells that contain a gene trap insertion in the receptor will become resistant and will outgrow the others. We will then, because we know the sequence of the gene trap cassette, we can amplify by a PCR reaction where we have enriched for gene trap insertions, and we'll identify in an unbiased manner by deep sequencing where in the genome that is. And you see that by and large, we really only found one locus in the genome here on chromosome 19, I believe, that is the gene that is called LSR, and you know, this is the receptor for that particular toxin, as it turns out. And so, let me just show you one additional experiments that these people did to confirm this. So one experiment that I think is fairly impressive is that you take plain HeLa cells. Those cells, if you expose them to that toxin, will not really respond because they lack that particular receptor. The moment that you transfect the cells with that receptor, they become susceptible to the receptor. And then fair enough, you can show that if cells overexpress the receptor, you can actually take labeled toxin, and that will adhere to the cell surface of those cells expressing the receptor, but not to the control cells. So there are a lot more experiments in that paper, but all in all, this would show that we can identify resistance factors, that we can identify genes that, if knocked out, confer resistance to that toxin. And in that particular case, we actually uncovered the receptor of that toxin, which is the lipoprotein receptor, LSR. And so let me summarize that first part for you. And that first part was really about unbiased haploid genetic screens. And you saw the outcome of that screen was really a digital answer. It was one gene that conferred resistance. It's not multiple. It's not like an RNAi screen where you end up with a few hundred hits where then you're not sure what they do and how you need to follow them up. The screens reveal remarkable biology. There are a couple of published examples, and I'm going to share a literature list in a moment with you. The screens are performed at high genome coverage. They provide comprehensive answers, so usually what you see is really what there is to be discovered. Most importantly, the results that we obtained with more than 15 such published screens and probably more than 100 screens that we have access to confidentially is that the results that we obtain with those genetic screens and that we obtain in general with these KBM7 cells usually translate very well into other cellular systems. So we haven't really encountered a single case where we found a strong genetic dependency uncovered by such a genetic screen in KBM7 that if then translated into other cellular systems have not turned out to be true. And I think in many ways these screens demonstrate that there is a lot of potential in using these cell lines, KBM7 and HAP1. I didn't talk about that second cell line yet, for biological experiments. And with that, I would just like to highlight that this is the seminal paper that described the technology, and since then there were a number of papers in really renowned scientific journals, and they, I think they really make the point that this is a very powerful approach and really enables yeast-like genetics in human cells. So at Haplogen we felt that the system represented an incredible opportunity, and so we thought, why not do something very straightforward, and that is to start off with those cells, infect them with the retrovirus at a relatively low multiplicity of infection to make sure that we really only have one gene trap insertion per cell, fax sort the resulting cells to make sure that we don't isolate wild-type cells, and then prepare limiting dilutions to isolate clonal cell lines where every cell line would contain a single gene trap insertion, and because these are haploid cells, they would presumably be knockouts for the corresponding gene if that gene trap sits in a human gene. And so we went ahead and did so, and we ended up with what we believe is a very versatile toolkit that enables human cell genetics. We characterized about 50,000 clones in the meantime. We believe that this is the largest such collection in the world. It covers about 30% of the expressed genes, of the genes that are expressed in that particular cell line, more than 10,000 individual clones. I think it's attractive that these gene trap mutants have LoxP sites, and I'll talk about that in a minute in more detail, because it allows you to reverse the knockout, and then, of course, as a consequence, you expect a reversion of the phenotype. These are all barcoded, which I think is very attractive because it enables other screening opportunities, and they're made publicly available now through our collaboration with or partnership with Horizon. And so the applications that I can think of are numerous. The most, let's say, straightforward one is really a functional genetics approach where you can study individual genes. You can obtain, if you like, a knockout on demand and see whether in your whether any, let's say, hypothesis that you have on any particular gene is true, and you can test that using our knockouts. Of course, you can think of many other applications on top of that. First, I wanted to point out that these are barcoded clones. I think that has several advantages also for you in using those clones. So every gene contains a unique 22 base pair sequence that you can amplify with a very straightforward PCR and sequence with the Sanger sequencing protocol. First of all, these barcodes serve as clonality markers, which is attractive. Second of all, these barcodes help you identify whether you're working with the right clone. So if, for instance, we sent you a panel of 5 clones and you were not sure whether they were mixed up in your hands, or possibly in our hands, we don't usually do that, but that may still happen, right? Then you could PCR up that barcode and make entirely sure that you're dealing with the right clone. And on top of that, we would like to use the barcodes in future to actually do genetic screens in those cells based on the barcodes, much like people have done in yeast. All of our clones are validated in great detail. And so typically the way we validate the clones is if a gene trap insertion lies, say, between exon 1 and exon 2 of a gene. We place primers here and there, so one primer in exon 1, one primer in exon 2, essentially flanking the gene trap insertion. And we would then perform qPCR. And so in wild-type cells, we expect a qPCR product that, of course, should not be present in the gene trap mutant cell line. And that is an example of just one clone, where you see that clone, these are duplicates, so you see the 2 curves that amplify very nicely in wild-type cells, yet in the mutant clone there is no signal anymore because the gene trap disrupts the gene expression. So we do that kind of validation for every clone that we send out. So should you be interested in any clone that we have, we'll actually thaw that clone up, we'll design specific primers for that particular clone, we do that type of qPCR reaction, and we report the results to you, and you're only obliged to purchase that clone once it fulfills our quality standards. Well, if you then look, let's say, at a few clones, at a few selected examples, you'll appreciate that whenever you have a gene trap insertion in a given gene, you'll see a reduction in transcript levels. So this is an RT-PCR, it's non-quantitative, but here is done for 10 different clones, looking at 10 different genes. And fair enough, you see a similar picture if you look on the Western blot level. So here we're looking again at around about 10 clones and around about 10 antibodies. And of course, you know, the beauty here is really the specificity, right? Because the gene trap has a very localized effect, and of course it does not affect any other genes in the genome. And we can actually, I think this is actually really a distinguishing feature of these gene trap mutants, that because we have the sequence of the cassette and because this cassette is a barcoded cassette, we can make sure that we really only have one cassette insertion in our cells, and it has a very specific effect that is very local, and we can exclude really off-target effects, which are problematic, as you know, with RNAi, but also with the recently published CRISPR-Cas9 system. Well then, our gene trap mutants have LoxP sites. Most of them have LoxP sites. We have different, let's say, generation of gene trap vectors that were used, and so our clones come in different flavors. Quite a number of them have parallel LoxP sites, and so if you transduce those cells with pre-recombinase, what will happen is that essentially the intervening sequence or the gene trap will be excised, and your knockout will become wild type again. We have anti-parallel LoxP sites in some clones where the cassette will then be turned. However, the disadvantage, and that's why we only have quite few clones with that LoxP configuration, is that multiple rounds of inversion are possible. And then we have so-called paired incompatible LoxP sites that really only flip the cassette once, and those will actually come handy also for another set of clones, which we call the inducibles, but I'll talk about those in a minute. So essentially, just to summarize that part, what these LoxP sites enable us to do is, whenever we have a clone and we see a phenotype in comparison to wild-type cells, we can take that clone, we can transduce it with CRE, and we expect that the phenotype that we see in that knockout clone reverts back to wild-type, right? So it's a very nice control that enables you to assess that the phenotype that you see is not due to genetic drift, because we're looking at a clonal cell line here, but is really due to the gene trap insertion that you have. I'm showing you one example here where we excised that cassette in 3 different clones. And so first you can check whether the excision happened at the level, at the genome level. And so if you place primers here within the gene trap cassette, you expect that PCR product to shorten, and that is pretty much what we see for all 3 clones upon transduction with CRE. And if we then look at a clone and we pick the p53 knockout we have, you see that it's actually quite a nice knockout on the protein level. There's p53 expression in wild-type cells. And the moment you take the knockout and you reconstitute it, or you introduce CRE recombinase, don't reconstitute it here, you introduce CRE recombinase, you see that wild-type p53 expression is restored. Let's look at a few phenotypes, and that should really highlight how those cells can be used to study individual genes that you may have under investigation. We picked here a few examples of well-established genes to be sure that the phenotypes that we get correspond to the phenotypes that were reported in the literature. But of course we believe that the potential is not really or not necessarily or not limited to studying TNF signaling, where a lot of tools are already available, but it really lies in the areas that are less well studied, where less tools are available. So here we're looking at cells that were stimulated with the cytokine TNF-alpha that exerts its action through the TNF receptor 1, and if TNF stimulates its receptor, it will lead to degradation of the inhibitory molecule I-kappa B. And that is pretty much what we see in wild-type cells here on the left. So if we treat those cells with TNF over different time points, we see that I-kappa B is degraded. However, in cells in which we have a gene trap insertion in the TNF receptor, I-kappa B is no longer degraded and is fully stable. The second example I would like to share with you relates to TRAIL-mediated apoptosis. So TRAIL triggers apoptosis through a molecule that's called caspase 8. And we see here that our cells, wild-type cells, or control cells in the white bars, with increasing concentrations of TRAIL, they succumb to apoptosis, yet the caspase 8 gene trap mutant that we have is completely resistant to apoptosis induced by TRAIL. Yet this is a very specific phenotype. So we're looking here at a comparison with where we treat cells with doxorubicin. Doxorubicin would also trigger cell death, yet cells lacking caspase 8 are still susceptible to that particular cell death pathway, so it's specific to the TRAIL-mediated apoptosis pathway. The third example I wanted to share with you relates to a pathway that is important in providing GPI anchors to certain molecules that then end up on the cell surface. And so this GPI anchor is a very complex structure that needs to be assembled with the help of a number of human genes, and those genes have these abbreviations. PIC-S and PIC-X are 2 of them. There's a number of others. And so we wondered if GPI anchoring is defective in cells having a gene trap insertion in the PIC-S and PIC-X gene. And the way we assessed this is we looked at CD59 because we knew that this was a gene that is localized to the cell membrane by the means of a GPI anchor. And what you can nicely see here is that in control cells, CD59 is displayed on the surface, yet in PIG-S or PIG-X mutant cells, we no longer observe it on the cell surface, presumably because the entire GPI anchoring process is really disrupted as a consequence of the gene trap insertion in those two genes. And so we believe that these three examples illustrate how our clones can be used to study the genetics of a particular phenotype, and we believe that they're very versatile and that these cells can actually be used for a lot of things, certainly for a lot of things that go much beyond, let's say, the biology of blood cells or the biology of leukemia cells. So pretty much anything that can be studied in those cells, we can provide you with the genetic tools to work out the underlying genetics. There is a last set of clones that I would like to mention that we also have. So these are clones that have the gene trap in an inactive configuration. So they contain the gene trap in the inverse orientation. I didn't mention this before, but the gene trap really only works if inserted in the correct orientation, and that is because the splice acceptor has a certain 5' to 3' directionality. And so in those cases, we can actually use pre-recombinase to flip the cassette, and once that cassette turns, it will actually result in a knockout, much like I described to you earlier for the mutant clones. And because these knockouts are inducible upon addition of pre-recombinase, we call them the inducible clones. And so these, of course, we cannot validate in the same manner as we do with the mutant clones, because if you like, they contain a sleeping gene trap that by itself is not active, and that would have to be awoken in your own labs by addition of pre-recombinase. And so the way we would typically validate those cell lines, those inducible cell clones, is that we look at the genomic level whether we have a PCR product in wild-type cells that disappears in the clones, much as it is shown here for two inducible clone examples. And so we have that product line available as well. Those clones, because they need an additional, they're not ready to use, right? The mutant clones are ready to use. They come, you can draw them up, you can work with them, you can study your phenotype of interest. Here you need to invest a little bit of work by transducing them with pre-recombinase, and so we felt because that additional extra work was required, they come at a discount. And so with that, I would like to summarize the second part. We believe that we've assembled the largest collection of isogenic human knockout cell lines on the planet that enable yeast-like genetics in human cells. Those clones are validated with great care. They're ready to be used. They come, you thaw them up, they work. Many clones are reversible upon addition of pre-recombinase. We believe that the results that we obtain in KDM7 cells in general, and as a consequence also with our clones, usually translate very well into other cellular systems, and we believe the most straightforward application that we see for those clones is what people call reverse genetics. So typically these cells come handy once you have a question like, does gene X or my gene of interest have an impact on phenotype Y? And with that, I would like to close. I would like to point out that this collection has been, the first draft of this collection has been published in Nature Methods in October last year. This is a public-private partnership between Haplogen and the Center for Molecular Medicine of the Austrian Academy of Sciences. I leave my contact information here.
All right, so first off, why did Horizon want to partner with Haplogen? Both organizations are very, very focused upon customer value, and specifically in the area of translational genomics. One of the biggest challenges around determining the gene function of specific genes is the ability to actually knock them out. And in a diploid model, that can be challenging, even with the best gene editing technologies. And that's why a haploid model can be really quite attractive. And it's as a result, the number of validated cell lines that currently exist is fairly limited. At Horizon, we have over 550, and we believe firmly that we have the largest catalog of isogenic cell lines in existence. But still, when you consider the sheer volume of genes and all of the genotypes that can exist out there, there's a lot that we haven't covered. So as a result, the attractiveness for haploid cell lines is really quite clear. So these represent truly biologically relevant in vitro models for your experimentation. So the offer itself and how we're providing these to customers is through a licensing model. Clones are ready for shipment within 6 weeks. They're validated upon order. If you're a commercial entity, you can receive a license for, the standard is 1 year, but there's some variability. If you require a longer license, then that's certainly, we can certainly discuss that, and there are discounts available for that. And then you can see in the table there what the rough pricing is. Of course, all of this information as well is also available on the Horizon website, and we encourage you to take a look. There's additional brochures and more material there as well that we'd encourage you to take a look at. If you're an academic, license is for an unlimited term, and that's at a significant discount. And Tillman had mentioned about how they're inducible clones, and those are with the gene trap and the reverse orientation. Those are provided at a 25% discount.